puzzled about, all right, now it's being recorded. Okay, so I have spent about 3/4 of my time in the past 10 months attempting to set up a COVID testing facility here, which can run about 100,000. Well, the goal is to run about 100,000 PCR-based tests per day for a marginal cost, a staff of about 12 and a marginal cost of about $3 a test. Anybody that's interested in that kind of stuff afterwards, I would be happy to chat about it. It's all I've been thinking about pretty much for the past year. But there's really not any science to it whatsoever. It's all about like supply chains and FDA regulations and so on. And so, although I'd kind of like to talk about it, I felt like it was inappropriate here. So instead, I'm going to talk about sort of standard stuff, work that we've been doing over the past few years, mostly on hold over the past year. But as many of you know, I've been interested in understanding, essentially how evolutionary dynamics acts as a sort of stochastic algorithm to take us from some set of possibilities, some fitness landscape in any particular scenario, to the kinds of evolutionary outcomes that we observe, the patterns of variation and so on. And so what I'm going to talk about today is just experimental methods that we have developed over the past couple of years, and some published and some unpublished work using basically high efficiency DNA barcoding to track evolutionary dynamics in laboratory populations. So before I get into the newer stuff, I just want to briefly recap the kind of system that we're talking about and the way in which we would observe evolutionary dynamics in these laboratory populations before we started getting into all of this barcoding. So, the canonical laboratory evolution experiment is Rich Lenski's long-term evolution experiment in E. coli. Most of you probably already know about this. And basically, Rich has been maintaining 12 replicate populations of E. coli in a batch culture protocol, basically diluting them by a factor of about 100 every 24 hours for 32 years now. Well, at least up until last March. They're currently in the freezer. So he's been maintaining these populations, as I said, 32 years. And this sort of batch culture protocol corresponds to an effective population size of roughly 30 million. And a few years ago, we got interested in tracking evolutionary dynamics in this system using sequencing. At that time, there was about 60,000 generations of evolution that had been completed. This was as of about 5 years ago. Now they're up to about 70,000 generations. And Rich had frozen down samples since 1988 at 500 generation intervals. And, in this system, there's a lot of complicated things going on. That's the sort of time scale, a lot of complicated things going on. One can make a sort of crude measurement of fitness. I mean, for whatever it's worth, there's all kinds of complications to this, which I won't really get into. But by competing evolved strains against the ancestors, one can measure the rate at which evolved strains isolated from different time points over the 60,000 generations of evolution, the rate at which they outcompete the ancestral population. And that's a sort of measurement of the population average fitness difference between that evolved isolate from that particular population in that particular time point relative to the ancestor. And what you see is a kind of rapid increase in fitness early in the experiment, followed by some sort of long, steady increase. And this is very typical of laboratory evolution experiments in E. coli and other systems like yeast that we do in my lab. And the sort of question that I've been interested in over the past half a dozen years is how we can look at what's going on at the sort of molecular level underneath these sort of slow, steady, fairly repeatable increases in fitness, gradual increases in fitness over time. And so the way that we used to do this a half a dozen years ago is by doing sort of whole population metagenomic sequencing through time. So for example, we took all 120 samples, basically at 500 generation intervals from the 60,000 generations of evolution in this long-term evolution experiment. in each of the 12 replicate lines and did whole population sequencing. And the kind of thing that you see when you do this is that you can identify novel mutations that arose spontaneously during the evolution of each line. And based on the frequency of those mutations in your metagenomic sequencing, you can track their frequency through time. And so the sort of thing that you see is that mutations arise. In this particular population, by the 500 generations of evolution, there were a half a dozen or so mutations that had spontaneously arisen and come to some appreciable frequency, 10%, 20%, in this case, 50% frequency within the population by that time. And then you can sort of track what happens to them after 1,000 generations. Some of them have gone to fixation, others have gone extinct, new mutations have arisen, and we see this sort of dynamics continue through time. This is the first 10,000 generations of one of the populations. And if we look across all replicate populations, here are 6 of the replicate population, 6 of the 12 replicate populations. And you see, all kinds of interesting evolutionary dynamics where lots of different mutations are arising. They're competing with one another. In some cases, as you can see down in these bottom populations, as well as the one at the top, very clearly, there appear to be spontaneous emergence of ecological interactions where you have coexisting strains that persist for tens of thousands of generations, even while new mutations are constantly arising within each clade. If I look at the the other 6, we see these kind of even more wild dynamics where in these 6 populations, at some point, a mutated allele arose and spread through the population, and you have a sort of wall of color because there are thousands of mutations that arise in these populations. And so, there's a lot that one can say about evolutionary dynamics from this kind of data. But the fundamental limitation is that because of practical limitations on the sequencing depth at which you can achieve, as well as limitations imposed by the error rates of Illumina sequencing, we can only see mutations when they reach an appreciable frequency within the population. I mean, depending on how much money and effort you're willing to throw at it, you could maybe get down to 1% or a fraction of 1% frequency. Here in this particular experiment, we see mutations only when they rise to somewhat higher frequency, 5 or 10% of the population. So that really is only the tip of the iceberg, because as you may recall, these experimental populations, they have population sizes, in this case, of 30 million individuals. And so by the time you reach 10% frequency, you've already gone from a frequency of, on the order of 10 to the minus 7 up to 10 to the minus 1. And we're seeing that sort of last order of magnitude of frequency dynamics, but we don't get to observe what happens to these populations at much lower frequencies. And so to address this question, basically, we set out to basically generalize an approach that was first introduced by Sasha Levy and others at Stanford, particularly Gavin Sherlock and Dmitry Petrov and Daniel Fisher's group and Jamie Blundell, I think, pioneered some of the analysis methods for these kinds of approaches. And the basic idea is that what you want to do instead of whole genome sequencing is to introduce DNA barcodes into evolving populations that basically label small sublineages within that population. Ideally, you would uniquely label every individual cell with a unique barcode. You don't really quite do that, but you can label very small sublineages of the population with unique barcodes. And then you evolve for a little while. And some of the barcodes, there's some genetic drift process that goes on, as well as new mutations arising. And some of the sublineages get beneficial or deleterious mutations and increase or decrease in frequency. And based on those barcode frequency dynamics, we can infer the mutational dynamics that are driving the evolution of the population. The problem with the first version of these approaches is that what happens is that some lineages get beneficial mutations and they start to spread through the population and they take over. And before too long, basically when a selective sweep has occurred, you no longer have any barcode diversity in the population and you sort of become blind again. And so a graduate student and a postdoc in my lab, Jose Rojas Echenique and Alex Nguyen Ba, developed a method to sort of repeatably barcode evolving yeast populations. So basically, you introduce DNA barcodes, you allow the evolution to take place, and then you sort of subdivide the lineages by adding additional barcodes sort of immediately downstream of the next one. And you can sort of subdivide each of these lineages into sublineages and track their dynamics forward, and then subdivide again and again and again as you move forward as the population evolves. And so there's a sort of complicated technical details to how they achieved this using a sort of Cree-Lock system where you basically integrate successive barcodes one after the other by using a system of 3 orthogonal types of lock sites and a sort of cycling marker system, which I don't want to get into the details of this in the course of a half-hour talk, but anybody who's interested, I mean, this is now published, this system, and there are various sort of updated versions of it, which I'm happy to tell people about, anyone that's particularly interested. But the bottom line is that you get basically after many subsequent rounds of barcode addition, what you can do is you can sequence a whole population, and each individual you'll get a sort of will have a sequence of barcodes that encodes its entire ancestral history. And so what this kind of looks like is like this, okay? So we start with a population, okay, at some initial time point where you've got a whole diverse set of barcodes, and there's so many of them. We have, on the order of 10 to the 5 barcodes in this unique barcodes in this population. So they're all at such low frequency, sort of subdividing the population into tiny little lineages that I can't even see any of them. And then a whole bunch of mutations start to arise as the population begins to evolve in this environment. And genetic drift is also, of course, occurring. And some of these lineages start to increase in frequency due to acquiring beneficial mutations or potentially due to genetic drift. And what we start to see is some of the barcodes reach a large enough frequency that we can see the lineage that has that barcode. And what we can see here is this sort of pink lineage will eventually sweep through the population and take over. But at this generation 100, what we've done is we've added an additional barcode, which basically subdivides all these lineages. And you can see at that point, this pink lineage was pretty small. And so it got subdivided into a dozen or so sublineages, which all, then later on collectively sweep through the population, but individually they're subsets of this lineage. And that happens to all of these other lineages. The same thing happens again. We add sublineages at generation 200. Here you can see, right, there must be additional beneficial mutations that happen to occur. For example, in this, can you guys see my cursor? I can never tell on this. Maybe hang on.
Right now, I cannot see your cursor.
Hang on, I'm gonna do this. I should know this by now, right? Okay, so now you can see.
Yes, it just appeared. I mean, yes.
Yeah, so like, for example, this sub-lineage has acquired some mutation or maybe done very successfully due to genetic drift and is now expanding within this background that had this initial barcode. And we continue this through time. And essentially what we're doing is we're sort of keeping track of something not quite, but approaching the sort of complete phylogeny of this whole population as it evolves. And we're seeing basically how many offspring each lineage and sublineage and sub-sublineage and so on has through time. And so based on that, basically what we did was we did a bunch of different populations, okay, in different environments. I'm going to talk about a few of them. So here are a couple of populations evolved in a sucrose environment. Here's a couple of fluctuating environments at the bottom, which I'll talk about briefly at the end. What I want to focus on to begin with is just looking at how we analyze these lineage dynamics using two sort of populations as examples. So this here at the left is evolution in a rich media environment, YPD. This is sort of rich laboratory media for yeast. And here is adding a bit of stress, acetic acid to the media for the population at the right. Okay. Now, what I'm showing you here is this sort of crude lineage dynamics, sort of in the same way that I was showing you on the previous slide. So basically what we need to do from this kind of dynamics is to understand, what aspects of this are driven by genetic drift, and to what extent can we infer new mutations that have arisen and how they're competing to drive these lineage dynamics that we see here. So a graduate student at the time in my lab, Ivan Asfievich, developed a statistical method to do this. And basically, the idea is that you have hundreds of thousands of barcoded lineages and sublineages that are competing within the population at any given time. And the vast majority of that dynamics is just driven by random sampling and genetic drift. And mutations are relatively, I mean, they're important, but they're relatively rare, at least at the level of individual sublineages that consist of a few cells. So we can use the overall lineage dynamics to construct a sort of null distribution of the properties of genetic drift in this particular system, just empirically from the data. And we can then sort of discard all of the dynamics that are just reflective of that genetic drift and look for lineages that sort of expand or contract in ways that cannot be explained by that null model of genetic drift. And so we group lineages that do that. And then essentially what we're looking for, we have sort of, lineages at all levels of the hierarchy of barcoding. So we have, parents and children and grandchildren and great-grandchildren and so on. And we essentially identify when and in which lineage new mutations arose by looking for lineages essentially that expand, that grow or shrink in a way that's sort of different from their siblings, but is inherited by all of their offspring. And so that allows us to identify new mutations, essentially, that arise, and they're, the new selected mutations that arise. And so based on this, basically, we can take all of this kind of lineage dynamics that we observe and sort of infer when new mutations have arisen and their frequencies as a function of time. And based on the way in which those mutations, they increase in frequency or they decrease in frequency, we can infer their effects on the fitness of the corresponding lineages. And so that's what we're sort of seeing here is each of these stars is a new mutation, which we have inferred to have arisen at some interval here. These white strips are the actual evolution, and these gray areas are the kind of points at which we added new barcodes. And you can see that that imposes a certain perturbation on the system. So really, like, this is actually kind of looking at evolution to some extent in an environment that's fluctuating with time, which I'll talk more about in a few minutes. But what you can see is that I've highlighted some of these lineages with different colors. So for example, this orange mutation, it's sort of growing through the population, and then it gets sort of submutations, sublineages get mutations, some of which sort of grow and take over within that orange group. And the way that we sort of visualize then how this competition between lineages and sublineages takes place sort of goes back to these theoretical models of evolution in these kind of laboratory experiments, which models the evolutionary dynamics as what we call a traveling wave. And so basically the idea is that you have a large population where you've got lots of different mutations which are sort of competing and interfering with one another. And the basic picture that you have is that sort of collectively, all these different mutations, they generate variation in fitness within the environment, within the population, which I'm sort of drawing by this sort of blue diagram. And then each new mutation we can think of as sort of arising in some background, generating some new mutant lineage, which is the combination of the sort of background in which it occurred, as well as the effect of that particular mutation. And then that background competes against the sort of collective effects of everything else. So for example, this is a sort of modestly beneficial mutation that occurred on a better than average genetic background, generating a highly fit lineage that started to increase in frequency. And then over time, it sort of increased in frequency, but all the other lineages in the population eventually caught up and overtook this lineage. So then it eventually went extinct. Or alternatively, the lineage could sort of continue to get new mutations and sort of surf ahead of the rest of everything else. And that's how a lineage would eventually fix. And so what we can actually do with these kind of barcode dynamics now, where we can identify mutations when they happen at really low frequency and sort of track their interactions, is actually visualize this whole traveling wave in the experiment as it happens. And so what I'm going to play for you is just sort of one example, that YPD population that I was showing you earlier, and the traveling wave of fitness as the competition between different lineages arises. So initially we start from some clonal population at time 0, and then I'm just going to play this, and time in generations is sort of moving on. New mutations are arising here, all these different sort of colored lineages. The mean fitness of the population in black is sort of increasing with time. And you see this sort of traveling wave, let me play that just one more time, of lineages of fitness as it sort of moves on, and the sort of way in which all of these mutations kind of compete with one another within this wave. And another way to sort of visualize this, see if I can do this, hang on. Oops. Is sort of like this. If I'm looking at the sort of fitness axis on here and time sort of going back, what we see is, initially you have some clonal population, numerous new mutations sort of arise. They start to increase in frequency, but collectively that sort of increases the sort of average fitness of the population. That's this sort of black line. And some lineages kind of get left behind and go extinct, while new mutations kind of are constantly arising. And the sort of fate of each lineage is determined not by sort of some individual mutation, but by the sort of, this kind of jumping and surfing process. So this sort of gives a sort of concrete experimental system in which we can look at these, dynamics of traveling wave adaptation in microbial populations. We've also sort of started to look more explicitly at more complicated systems where, for example, environmental conditions fluctuate over time. And this is something where, I think these same kind of theoretical ideas about traveling waves of fitness can be generalized to understand how specialist or generalist phenotypes may evolve in fluctuating environmental conditions. So I'm just going to show an example of a couple of situations where we have fluctuating environments. We can do the same exact kind of thing where we infer when new mutations arose and their fitness effects in each environmental condition. So these two environments we chose, they're just fluctuating between the two conditions that I was talking about earlier, the rich media and the somewhat stressful media. And we just alternate every interval, every 100 generations, we switch between the rich media and the stressful media, and then back and forth. And so there may be some mutations that help you in both conditions, some that help you in one but not the other. And we just want to see what happens as these populations evolve. And so I'm just going to sort of play for you what happens. Okay, and now what I'm sort of going to show is we start with this sort of clonal population that has this sort of ancestral fitness in both conditions. And for 100 generations, we're going to kind of select in this environment, so this arrow, and then we'll switch back and forth and back and forth. And what we'll see is new lineages. So here, for example, are new mutations arising that confer some increase in fitness in this stressful environment on the vertical axis, and actually slightly reduce fitness in this environment represented on this axis here. Okay, and we're going to see the size of the clones corresponding to those mutations by the size of these dots. okay? And I'm just gonna take a minute to sort of play what happens, you know, as we evolve, switching back and forth between the way that we select in these two environments. And you can see in black is the sort of mean fitness of the population in the two environments. In this particular case, we seem to be getting some sort of generalist adaptation overall, where you get better in both conditions, okay? Although clearly there are sort of mutations that tend to take you up in one direction and down in another, or vice versa, okay? We see, just to play, here's the other replicate population, okay? This is exactly the same experiment. It's just a different replicate population, okay? And what you're seeing is, you know, we have the same sort of, or at least no evidence for any sort of different sort of joint distribution of fitness effects of mutations, right? There's still, you know, mutations that are good in both, some that involve trade-offs, but yet this population seems to do something, you know, somewhat different simply because of the order and the timing with which we get these different kinds of mutations. And so I think like this is a further direction for theoretical work to understand how, you know, the timing of the fluctuations in the environment and the joint distribution of fitness effects across the two environments leads to these kinds of dynamics where we can either get generalist or specialist phenotypes with different probabilities given different population parameters. And this sort of experimental system, I think, helps you visualize the kind of processes that we'd have to think about in such modeling. So I want to just stop there because I think I've talked for 20 or 25 minutes or something, and leave plenty of time for questions. There aren't really any sort of firm conclusions. I just think it's sort of neat to be able to sort of see these dynamics at this kind of unprecedented resolution, given these barcoding methods. And this is a picture just to acknowledge folks that have really led all of these kind of experimental developments. Unfortunately, our last retreat was supposed to be in April of 2020. So unfortunately, I have to give you the several years old picture because obviously that didn't happen. But all this work was led primarily by Alex Nguyen Ba, who just recently started his own lab at University of Toronto, Jose Echenique, who is now a postdoc also in Toronto, Catherine Lawrence, and Ivan Asfijevic, who is now a postdoc at Stanford. I'll stop there and get with any questions.
Okay, thank you very much, Michael, for the great talk. So we have a few minutes for questions indeed, so feel free to ask questions either in the chat or by speaking up. We already have a question in the chat from Tamara. Tamara is asking, do you have an idea why the adaptation curve in the Lenski experiment has two phases, rapid and slow? Is it captured by the traveling wave model? Do you see it in your experiments?
So we do see it in our experiments. It appears to be a fairly sort of general feature of these laboratory evolution experiments. And it seems to arise from, at least in large part, a sort of general pattern of diminishing returns epistasis, where you have a sort of large pool of available beneficial mutations starting out. And as you sort of adapt, to a certain extent, there's sort of evidence for contingency where, you know, you close off, you know, you get mutations that help you adapt in one way and that sort of closes off other pathways to adaptation, although it in some cases opens up additional opportunities. But overall, there's a sort of general pattern where the same set of mutations becomes sort of systematically less beneficial as you get more and more fit. And in fact, you also sort of see a pattern where some mutations, you know, that were maybe weakly beneficial start to become deleterious as you adapt, and weakly deleterious mutations become increasingly more deleterious as you get more and more fit. And, you know, it's unclear exactly why this pattern of diminishing returns exists, although George Zhang's lab published a nice paper sort of proposing a general mechanism for this. And I there's a preprint that my lab posted. Gautam Reddy is the lead author on that, proposing a similar sort of explanation. But this sort of general pattern of diminishing returns appears to be, at least in large part, responsible for this kind of slowing down of adaptation over time.
Okay, great. We have another question from Vera Kovakova. Do you observe sequencing or ligation biases favoring some specific barcodes? How do you control preferences towards certain barcodes?
Yeah, I mean, there are all, I mean, I could give a long and very boring talk on all of the technical issues associated with this barcoding thing. You know, we sketched it out on the board, on a whiteboard. It seemed like so simple. It took like 2 years to really get it to work. There are all kinds of problems. There are all kinds of problems which have all kinds of very, very detailed technical reasons. And we sort of controlled enough of them and made it sort of clean enough that we could get a reasonably unbiased dynamics out after several years of work. I don't know how to answer the question without really getting into technical details, which I would rather not. But I would be happy to sort of, the basic answer is, yeah, we see all kinds of technical issues and biases that we've done our best to control for, to eliminate experimentally where possible and control for computationally when we can't. And there's about 100 specific examples.
Okay, thanks. We have another comment and question by Eric van Nimwegen. He's saying, very nice. All this seems to nicely fit with the theory, right? So what's the most surprising thing you've seen?
I mean, I guess I wouldn't say it's necessarily surprising, but I mean, actually probably perhaps unsurprisingly, right? I mean, it does sort of in a qualitative sense fit with theory, right? I mean, you see these traveling waves, you see the competition between lineages, you see these kind of surfing dynamics of clonal interference and hitchhiking. And, you know, in a sort of conceptual way, it does sort of fit the theory. You know, for various sort of reasons of mathematical convenience, the theory, of course, as you know, you know, makes assumptions that we're in certain types of parameter regimes where, like, for example, we can sort of treat the collective dynamics of everything going on as leading to a steady state sort of fitness profile, that sort of blue wave that I showed in the schematic diagram. And we sort of, you know, assume that sort of has some steady state shape, and that we can sort of treat that steady state, that sort of average shape as constant and sort of ignore fluctuations in that. And, you know, you really see quite dramatic violations of that in the experiments. And so, you know, you get occasionally large effect mutations that lead to temporary collapses in diversity, which then get sort of reestablished later. You see, so you see things that sort of violate a lot of the practical assumptions that people make in analyzing the theory, and you know, it sort of points to the need to sort of think about fluctuations in the wave in, you know, making.
Hey, Mike, yeah. Thanks. So I was thinking of sort of more dramatic things like, you know, evidence of frequency dependence in fitness or so, things like that, right? I mean,
yeah. So, I mean, a few of the things that we hoped to get at with these traveling wave with these barcoding methods is frequency dependent selection, particularly acting at really low frequencies, potentially maintaining lineages at, you know, a frequency of 0.1% or 0.01%. We hope to see some sort of non-transitive interactions potentially between lineages. And we hoped to be able to see cases where, you know, you might have lineages that get beneficial mutations that then tend to sort of make them more adaptable or less adaptable, maybe by either through changes in, you know, subtle changes in mutation rates, or potentially changes in the spectrum of mutations that they can get going forward. We don't, in these experiments, have any evidence for any of these three effects in these experiments. Now, of course, you know, it's just a small number of experiments in a select, you know, few environments over a relatively short period of time. So, you know, if you did more, if we looked at longer time periods, hopefully we begin to see that. But we don't actually over these experiments. At least we see no convincing evidence for it, and we have looked.
Can they mate or not, these guys?
These are, I mean, they could mate, but they don't in these experiments. We have played around with, maintaining sexual yeast populations with these kinds of systems. If you think about it, there's all kinds of issues there. But you could, in principle, do that in a variety of ways, but we haven't.
Okay, great. I think in the interest of time, we should, stop the discussion here. Thanks again, Michael, for this great talk. And now we welcome our next speaker, who is